the higher vertebrates, Drosophila limb gland also getting the same type of response. But what is not known, which is the role of hematopoietic niche, or we call the microenvironment. In the mice, it is not known. And then in the fly also, then we decided, why don't we go to check the role of hematopoietic niche, because it is always good when we are using the model system. Uh, this is we have to give some new information which is not available. So this all this data which is proven, we can use this model for the, at least for the benzene induced hematopoietic response. So that we decided, why don't we understand the role of hematopoietic niche, which is not known right now in the case of benzene exposure. So what we did, on this is like hematopoietic niche fly also have the two type of niche. This one here, you can see this, because endothelial niche, this is vascular niche. The same type of niche is also present in the bone marrow also. So this is like functional and structurally they are the similar structure. So then we decided to investigate the role of hematopoietic niche or microenvironment in the benzene-induced hematopoietic response. So first we check whether there is any structural damage here. So after the benzene exposure, we did not get any structural change in this in this hematopoietic niche. Then we think maybe sometime hematopoietic niche also work in a non-autonomous manner. So what we did, we removed this niche by what genetically, because fly give this opportunity or this flexibility, we can modulate the fly in, like, we can overexpress, or we can delete any gene. So what we did, we just removed this hematopoietic niche, and then we expose the gene, then we check whether we are getting the response or not. So when we did this, here you can see, so this is the normal niche, and here we introduce the pro-apoptotic gene in the niche, and then there is no niche in this larvae. And then when we expose with the benzene, and we got the rescue. So here you can see, without, if niche is there, we are getting the aberrant differentiation of the blood cells. But when the niche is not there, the aberrant differentiation of the blood cells is rescued. Similar, the prolonged exposure, there is a tissue damage, but in this case, after the after removing of the niche, there is a rescue in the structural damage. So this prove, yes, niche is playing an important role in the benzene-induced hematopoietic response. So some signal this niche is giving to these progenitors. So we are interested what are these signals. So we did a lot of research, we did a lot of experiments also, and we came on one signaling pathway, which is like toll NF kappa B pathway. So basically, this is a immune pathway. Toll is a receptor, and this toll receptor is a what is once the ligand came, it's bind, and then it make a trimerization of mid tube and pelle, and then it start. Wait, it is some problem. Okay, so this ligand is coming, then there is a trimerization happen, then this degradation of cactus, then if this is a response, this will go inside, then it will start the activation of the immune related. So this is basically a toll pathway. Then we what we did, we just removed or blocked this toll pathway. And first we see what is the expression of this toll pathway. So we've got a toll pathway is highly upregulated after the benzene exposure. Okay, this is okay. In the PSC also, you can see it's a huge expression if you compare with the control. Then what we did, we removed this toll from the PSC or from the niche, and then similar, after removing this niche, we got the rescue in the after the benzene exposure or response. So we came to know, yes, in the niche, yes, this benzene is upregulating the toll in the niche, which is responsible for the aberrant differentiation and the limb gland dispersal. Okay. After then we came to know what is the mode of action. So mode of action, we know it's cause a lot of oxidative stress and toxicity. This is all known thing. So we checked whether ROS is there. Yes, after the benzene exposure, we got a lot of ROS. And then what we did, we quenched the ROS in the niche here. And what we found, yes, after the quenching the ROS, we found the rescue. So there are two things here. We are playing with the toll, we are playing with the ROS, and we are getting the rescue. So benzene is activate the toll activate the hematopoietic response, benzene ROS activate the hematopoietic response. So both we are getting the same type of. Then we ask the question whether toll and ROS are interconnected or not, or they are the same. They're the different pathway. So we did one experiment where we just quenched the ROS and we checked the expression of this toll pathway, and we got there is no activation of the toll pathway, which me and vice versa. What we did, we overexpress, we downregulate the toll pathway, and then we check the expression of the ROS here. So in this case, we did not get any rescue in the ROS production. So what we came to know with this experiment, yes, benzene is producing the ROS, and ROS is activating the toll NF-kappa B pathway, and this toll NF-kappa B pathway is responsible for the aberrant differentiation of the blood cell and the limb gland dispersal. So, and then we came to know is this response is really important for the organism survival. That is very important. So we found if this toll pathway is activated in the niche after the benzene exposure, the survival is more. If the toll pathway is inactivated in the hematopoietic niche, we got the more death. So what we concluded, this toll pathway in the blood cell development is really important, not only for the correct immune response, but also the survival of the organisms also. So here we found a new candidate in the role of hematopoietic niche, which is the new information which is still not available in the mice. So this is our contribution to the benzene-induced hematopoietic response, and I hope this similar type of response is also be there in the mice also, which is needed, which still we need to study. So this work has been recently published in Free Radical Biology and Medicine, and this is my PhD student Clinton D'Souza. Now he is postdoc in University of Washington. So he published this paper in 2022. And I'm thankful for the director of our institute, Nuksar, and Dr. Sham Prasad, and from Dr. Shama Prasad, Manipal University, for their use of confocal microscopy for this work. And yes, Dr. Michel Crozeta, who supported us for giving a lot of flight talks. And yes, and for the financial, I am really thankful for the DST SERV for giving us like a lot of money to conduct this research. So thank you, ma'am. I think I'm on time. Correct, ma'am.
Hello. Thank you, sir. So very informative lecture and enlighten us about the toxicity and their aspects. Thank you so much, sir. Now moving forward, so I would like to call Professor Dr. Vinay Dubedi, sir, for the today's session vote of thanks, sir.
So can I stop sharing here?
Yes, sir, you can stop.
Okay.
On behalf of Department of Biotechnology, I would like to express my sincere thanks to all the presenters, invited speakers. Thanks for your presentation and the valuable thoughts and the knowledge that is shared with the students. And I hope definitely this will help the students
Manish, sir,
to progress their research in the respective fields of molecular biology, plant pathology, mycology, CRISPR-Cas technology, etc. Thank you very much. Thank you.
Thank you, sir. So now I'm closing the meeting, sir. Thank you, everyone.
Thank you,
Thank
sir.
you.
Thank you, sir. Thank you, all the
Thank-
speakers.
Thank you
very
ma'am.
much.
Thank you, sir. Thank you. Thank you.
Dr. Anurag.
Thank you, sir. Thank you, sir. Thank you. Sir.
Right.
So can we leave some now?
Yeah, sure, sir.
Okay. Thank you, sir.
Sir.